So MedBlock contains some of the most interesting material. You guys can turn the lights down if you want to see that a little better. But I cry, and I actually worked on this stuff all weekend, and I just cried because I only have a few days to cover this material. Maybe turn the front lights down, and maybe leave the back. What do you guys think?
All four go down, and that one that's...
Or they're on the side. Yeah, whatever works, but yeah, whatever.
There you go.
Yeah. Can you guys still see well enough to do notes?
Yeah.
And all that? Okay. Because if not, you know, we can keep the back ones on. Okay, cool. So when I teach the biology 211, I have a little over a month to cover neurology. So I spend a little over a whole month, you know, five weeks. So basically a third of the semester we spend on the nervous system and special senses, and I have to gut that down to less than a week for you guys. So there's some crying involved because I really have to gut things down a little bit, but still. All right, so let's go ahead and get started here and make sure that works. Oh, look at that. All right. Nice. It works. Yeah. All right, cool. We'll be a Jedi. We'll be neurology Jedi. Okay. So what I want to do is I want to start talking about the nervous system. And what I've done differently from the book is I've kind of combined some of the pathology into the anatomy and physiology. You know, where the book, it kind of starts at the A&P first, and then it goes into the pathology, the different diseases. I've kind of mixed things up a little bit for this first lecture or two, because the diseases become relevant at certain points in the discussion, so I'll just bring those up. But let's talk about the nervous system here. So this is the big picture of the nervous system. And as you guys probably know, it's the command and control center for all body functions. And it's somewhat analogous to like the central processing unit and the hard drive for a computer. There are some analogies that we can draw from that. Not robust analogies, but if you want to make an intuitive connection, that's kind of what the nervous system's doing. If I'm to be totally honest with you guys, we're not entirely sure of a lot of it, actually. And we'll talk about some of the problems, something known as the hard problem of consciousness. And it really is a very difficult thing to talk about. So there are three major functions associated with the nervous system. We know that the nervous system receives sensory information, okay? It processes that information and integrates it, and then following the processing and integration, motor information is sent out. Okay, so there's some sort of response to the sensory information coming in. Okay, and that in a nutshell are those are the high yield major functions that we see within the nervous system. Now there are two major branches or divisions of the nervous system. There's something known as the central nervous system, or the CNS, and that consists of the brain and the spinal cord. And then there's something known as the peripheral nervous system, the PNS. Okay, and these are all of the nerves that come off of the central nervous system. And the nerves can come off of the central nervous system at two general areas. And these are what we call the cranial nerves and the spinal nerves. So nerves that leave or come into the central nervous system directly in and out of the brain itself are known as cranial nerves. So these come out of the brain and they go through little holes called foramina in the cranium, and they tend to innervate. Innervate just means a nerve going to a certain area. An area has a nerve. They tend to innervate areas in the superior aspects of the body. So you're talking face, eyes, head, neck, in the very upper part of the chest. Okay. Now, the other way nerves come and go out of the central nervous system is via the spinal cord or the spinal column. And nerves that come in and leave the nervous system via the spinal cord are what we call spinal nerves. So here I have a cranial nerve that's leaving directly out of the brain, And here I have the spinal cord, and I have a spinal nerve coming off the spinal cord. You guys cool with that? Probably nothing you didn't already know, right? Hopefully. All right. Now, there are major pathways, major nervous pathways. And the two major pathways are what we call the afferent and the efferent pathways. Okay? And afferent, I just think of as ascend. Okay? So these are nervous pathways that ascend or move toward the CNS. So these are sensory pathways that brings information into the central nervous system. Are you guys okay with that? So afferent, ascend, sensory pathways. Okay? So this is information getting into the CNS. You guys cool with that? All right. And then there are efferent pathways, and these are known as descending pathways, and this is motor information. These are motor pathways that send information away from the CNS. Okay? So again, going back to the major functions, okay, sensory information comes into the nervous system via afferent pathways. That information gets integrated, and then there is a response. The nervous system wants to do something in response to that information coming in, and that information, that motor information, then leaves through efferent or descending pathways. Are you guys okay with that? So it's really sensory versus motor pathways. So far, so good? And then neurons, and we haven't talked about what neurons are, but they are the functional unit of the nervous system. Neurons that bring information between the afferent, efferent pathways, that kind of act as a bridge, are what we call interneurons. And they can bring information from one neuron to another. You guys cool with that? All right. So when we look at the major branches or divisions of the nervous system, everybody's okay with the CNS, right? The central nervous system. And what is the peripheral nervous system? This is everything else that comes off of the CNS, right? So these are the cranial nerves and the spinal nerves. You guys okay with that? Now, there are two major branches of the peripheral nervous system, okay? There is the somatic branch, or the somatic branch, and the somatic branch is voluntary. We have, for the most part, conscious voluntary control over this. And what do you suppose the major organ systems that are innervated by the somatic branch? Skeletal muscle, yeah. Okay. And then we have the autonomic branch of the peripheral nervous system. Okay. And I think of autonomic as being automatic. Does that make sense? So these are functions that are, generally speaking, outside of our conscious ability to control. We do have some ability to control some of these functions consciously, but these are pretty much automatic, and there are things that will automatically occur without us needing to consciously think about that. And then to further complicate it, the autonomic. Now again, the autonomic is a branch of the peripheral nervous system, but the autonomic is further subdivided into the parasympathetic and the sympathetic branches. You guys okay with that? All right. And then what I want to do is I just wanted to emphasize sensory pathways. So I wanted to emphasize a couple of sub-concepts underneath the umbrella of the afferent, or the ascending pathways. And these are what we call somatic sensory pathways versus visceral sensory pathways. So somatic, okay, again, soma literally meaning body, are sensory pathways that bring sensory information in from primarily the skin, okay? And then visceral sensory pathways primarily bring information in from the organs. Some of this information is we're consciously aware of. Most of the information we're not consciously aware of, okay? But that does give rise to different types of pain, okay? Specifically, somatic versus visceral pain. Have you guys ever heard of that term before? So what does somatic pain tend to feel like? Huh? Can be burning. What does it feel like when you cut your skin? It's a sharp, stinging sensation, right? What does visceral or organ pain tend to feel like, though? Dull, throbbing. Yeah. Okay. Diffuse as well. Anybody have an abscessed tooth? And that diffuse, throbbing, horrific pain, that's a visceral kind of pain. Whereas everybody's cut themselves, right? You've had a laceration and you kind of have that sharp kind of pain. And this is a result of sensors in different areas of the body sending information from the skin and from organs, and then that information gets integrated in the brain and it gets interpreted as being either somatic or visceral pain. You guys cool with that? Okay, everybody okay with some of the fundamental stuff, the foundational stuff? Cool. So let's now talk about the cells of the nervous system, and let's do this here. Bam, look at this. All right. Now we got our red lightsaber going on. Okay. So you have two different types of cells in the nervous system. You have what are called glial cells, or neuroglia, and these are the quote-unquote support cells. Back when I first took anatomy and physiology, there wasn't a whole lot of emphasis on glial cells. We learned, oh yeah, there are these glial cells and there's support and they kind of help form the blood-brain barrier and this and that. And then, that was, it was like 20 years ago when I did that. And now we know a heck of a lot more about the glial cells, and we know that they play a critical role in the central nervous system. And then you have what are called neurons. And neurons are classically considered the fundamental functional unit of the nervous system. And so this is actually a picture I took from Biology 211 of nervous tissue. Obviously, we've stained it to make it easier to see. But do you see these little star-like irregular structures here? You guys see that okay? Those are actually the soma, or the cell bodies of the neurons. And then do you see these tiny little dots here all over the place? These tiny little dots are actually the nuclei of the various glial cells. You actually have many more glial cells than you do neurons. But I want you guys just to think about this for a minute. You have approximately 100 million neurons in your brain, okay? How many stars approximately are there in our entire galaxy?
100 million?
There are approximately 100 million stars. So think about that. We have as many neurons in our brain as there are stars in our galaxy. This is kind of an amazing thought, and it's going to get even more amazing when we talk about the different connections that occur, but we'll get there when we get there.
Especially when you think that they're contained within this
I know.
tiny space.
Contain basically two hands, right? Make two fists, put them together, right? And that's a majority of your brain. And what's your brain weigh? About 1,500 grams. So a kilogram and a half would be a fairly large brain. So you're looking at 3-4 pounds of gelatinous matter, wetware, and that. That's actually a proper word. Wetware is biological. When we talk about a biological computer versus hardware, so wetware. I do want to spend a few minutes talking about the glial cells because they are important, and we tend to forget them in this whole everything's about the neuron discussion. So you have two major types of glial cells. You have your microglia and your macroglia. And your microglia are specialized macrophages. Remember, a macrophage is a monocyte. It's a type of white blood cell. And when the monocyte migrates outside of the blood, it becomes a macrophage. And these are phagocytic. What's a phagocytic cell?
They
They engulf. They eat mainly debris, cellular debris, sometimes pathogens and other organisms. So obviously these play a very important role in keeping the area around the neurons cleaned up, if you will. Does that kind of make sense? All right, good. And then we have what are called the macroglia, which tend to be larger glial cells. And then we have several different types of macroglia. I'm not going to be able to talk about all of them, but I want to talk about some of the major players. And the most abundant of the glial cells are what we call the astrocytes. These are most abundant, and they actually have little projections that link and hold on to the neurons and attach the neurons to the blood supply. And we think, some people think, others don't, there's actually some contemporary debate over this, but the classical story is that they help form something known as a blood-brain barrier. Are you guys familiar with the blood-brain barrier from prior classes? So the blood-brain barrier is essentially an additional membrane that separates the blood supply from the brain, okay? And this barrier presents unique challenges, particularly when it comes to pharmacology. Well, that's good and bad, okay? So you guys are probably familiar with dopamine, right? We've talked about it in pharmacology. But you guys probably know something about dopamine as a neurotransmitter, right? And you know that some diseases involve the loss of dopamine or the loss of dopaminergic neurons in certain areas of the body, right? This is a primary problem in a movement disorder called Parkinson's disease, something we'll talk about a little later on, right? And so wouldn't you think, well, if it's a dopamine problem, couldn't we just give our patient dopamine? What do you guys think? Yeah. It's very easy to give somebody dopamine, right? We can do that, IV, right? You guys all learn how to do it, and we know that its systemic effects act on vessels, on alpha receptors, and on beta 1 receptors in the heart, right? We know that. Why doesn't it improve things like Parkinson's if we were to give it IV? Because dopamine is a molecule that cannot get through the blood-brain barrier. So it is a way of separating the central nervous system from the peripheral nervous system. So it's a way of keeping stuff that goes on inside of the brain separate from stuff that's going on out in the body. Does that kind of make sense? And it also prevents some things from the body from getting into the brain. Now that can be good, right? Can help with toxins and infections and things like that, but it can be bad because it is exceptionally hard to target nervous tissue, to get drugs into the brain. This is why many types of brain neoplasms or cancers are very difficult to treat, aside from surgically removing them, because it's hard to get those chemotherapeutic agents into the brain itself because you have that blood-brain barrier. But we think that these astrocytes play a role in that, okay? They help protect the neurons. They are very important when it comes to regulating chemicals, okay? They actually have pumps that pump molecules in and out, away from and toward the neurons. They may help regulate the vascular tone as well, okay? The vasodilation and constriction. So very important. We also have what are called oligodendrocytes, and oligodendrocytes are important in that they form the myelin sheath of many of the neurons in the central nervous system. Now, this is only in the central nervous system, though. In the peripheral nervous system, the myelin sheaths are formed by what are called Schwann cells. So Schwann cells are the oligodendrocytes of the peripheral nervous system. You guys okay with that? Okay. Then you have something called the ependymal or the ependymal cells, and these are cells that produce cerebral spinal fluid. And why do we have cerebral spinal fluid? What's the role of that? What's its job?
Cushion.
Yeah, it acts as a cushion, right? And so basically your brain floats inside of your skull. Isn't that a weird thought? Your brain floats, inside of the skull. And in fact, the brain is so fragile that if you were to just take the brain unfixed without adding formalin and all that to it, but just take the brain and put it on a table, it would deform under its own weight. That's just how fragile it is. You guys all talked about suctioning, right? In some neurosurgeries, all they'll do, like if they want to remove a tumor or something, the tissue is so fragile that they could just hold a suction catheter up and it would start sucking. Yeah, it's very fragile. And so it makes sense that the brain floats surrounded by the cerebral spinal fluid, right? Because it would actually deform and could potentially become damaged under its own weight. Isn't that a weird thought? Yeah. You guys okay with that? So far, so good? All right. And then the satellite cells also are an important regulator in the external environment. So when it talks about the movement of ions and certain molecules across membranes, they play a role there. There's other types of glial cells, but these are the major players. Are you guys cool with that? All right, moving right along. Okay, I'm going to have to switch my little pointers here. Okay. So I just messed things up royally, I think. Okay, well. Okay, so this whole video kind of got messed up. Sorry, guys. I guess I have to stick with a laser pointer instead of the Jedi sword. Yeah. Okay. Let's see here. Well, maybe we'll be able to pick it up on the rebound. Okay. So you can see that the soma is kind of off by itself, and then you have the axon and the dendrites. And the pseudo-unipolar neurons are classically what we call sensory neurons. Okay? So your motor neurons tend to be multipolar, your pseudo-unipolar tend to be sensory, and then you even have what are called bipolar neurons, where the soma is right in the middle, and then you have your extensions that come out. And the bipolar neurons tend to be those interneurons, right? The neurons that interface between motor and sensory, or afferent, efferent. And then you have a fairly rare type of neuron called a unipolar neuron as well, but I really not going to talk about that in any detail. Okay, so you guys okay with that? Kind of makes sense? All right, cool. Let's move right along. Are you guys okay with that? The cell body. Now you have these little projections that come into the cell body, and these are known as dendrites. And dendrites bring information into the cell body where integration occurs. Are you guys okay with that? And so information comes into the neuron through these dendrites, and how does it leave the neuron? Through axons or through the axon. So information comes into the body, the soma, via the dendrites, and it leaves via the axon. Are you guys okay? Now this area here where the axon transitions into the soma is known as the axon hillock. It's a very important region when it comes to action potentials and depolarization of the neuron. So keep that in the back of your mind. And in many cases, the axons are covered in a myelin sheath. And what is the role of myelin? It basically is an insulator. It acts as an insulator, right? If you had a wire, okay, that was carrying an electrical current, and that wire was just kind of out on its lonesome, there would be a tendency for those electrons to kind of leak out of that wire, right? So what do we do commonly with wires? We insulate them, right? We wrap them in something that does not conduct very well. So something that, do you think it would be polar or nonpolar? You want something nonpolar, right? Polar things, right, tend to conduct, right? Because polar means that you have a separation in charge of electrons, right? So metals, for example, are polar, so you wouldn't want to wrap something in metal. Actually, metals make good conductors, right? So with that in mind, what do you think some of the most common non-polar molecules in the body are going to be? Fats. And so the myelin sheath is primarily made of a fatty substance. You guys okay with that? It's a lipid-based substance. Does that kind of make sense? And we'll talk about that in a little more detail. All right? And then you have these little areas in between the wrappings, okay, and these are known as the nodes of Ranvier, and these are important, okay, because there's a very different kind of conduction pattern that occurs down the axon, and it's known as saltatory conduction, and we'll talk about why that happens. And then you have this area here known as the axon terminal, right? And in the end, at the end of the axon, you have these little vesicles. And what is contained within these vesicles? Neurotransmitter molecules. And so when that action potential makes its way down, calcium channels open up here. Calcium causes exocytosis of these membrane-bound vesicles to occur, right? And then the neurotransmitters leave, okay, out of the axon terminals, and then those neurotransmitters act upon the dendrites of another neuron. Does that make sense? Or sometimes they act on other receptors. They don't necessarily need to be the dendrites of another neuron. They most certainly are in the central nervous system, but in the peripheral nervous system, they may be beta receptors, they may be alpha receptors, they may be nicotinic and muscarinic receptors as well as others. Are you guys cool with that? Okay, so the way that this whole action potential business works is it's an all or nothing thing. The neuron either undergoes an action potential, it fires, or it doesn't. Does that kind of make sense? And so there's a very Bayesian logic associated with this. And are you guys familiar with that term? Are you guys at all familiar with computers and how information is coded in computers?
Zeros and ones?
Zeros and ones, yes and no, on and off. Does that kind of make sense? And so there is somewhat of a parallel here with neurons. They either fire or they don't. Yes, no, 0, 1. Does that kind of make sense? So there is this sort of binary logic that seems to be associated with the neurons, at least in how they depolarize. Okay? Does that kind of make sense? Okay? So information or stimuli at the dendrites either makes the neuron more or less likely to polarize. This is a very important concept. So information, okay, that comes in and interacts with the dendrites will either make the neuron more or less likely to depolarize. You guys okay with that so far? Okay, cool. All right. Now, the little space in between the axon of one neuron and the dendrite of another neuron is known as the synapse or the synaptic cleft. You guys cool with that? If this synapse is not between an axon and a dendrite, but maybe the axon and nicotinic receptors of a muscle, a skeletal muscle, we don't call that a synapse, we call that an end motor plate or a neuromuscular junction. That's another name for a synapse of the somatic nervous system. Are you guys okay there with that terminology? Okay, so I want you to think about this. Each neuron may have axons that interface with approximately 10 to 20,000 other neurons. So think about this. One neuron may interface with perhaps 10 to 20,000 other neurons. So let's now think about this. So there are approximately 100 million neurons in your brain. Each one of these neurons interfacing with 20,000 other neurons. So there are 20,000 different connections between 100 million neurons. So if you go 100 million times 20,000, that's what, about 2 times 10 to the 12 connections, or 2 trillion connections within your nervous system. Isn't that interesting? And somehow, from all of these connections arises everything that we think of when we think of the human mind. Okay, at least that's how things are looking. And we'll talk about that in a a little more detail. Okay, so I want to talk about general types of neurons. So this is a classic example of a neuron right here that you see. And this is a very common type of neuron that we see in the central nervous system, but there are several types. So this is what's known as a multipolar neuron, okay? And this is a classic example of a motor neuron. A motor neuron tends to be multipolar, okay? So you have your soma, you have the dendrites into the soma, an axon that leaves the soma, and then several axon terminals there. You guys okay with that? There's also something known as a pseudo-unipolar neuron. And so what you have is you have a cell body here. The soma is not directly connected, not directly connected to the axon, but it is connected by a little extension here. So it's off on the side by itself. Do you guys see that there? And then you have... All right, so now let's get into the nitty-gritty of how the neuron depolarizes. So just like cardiac tissue, neurons maintain a membrane potential. Okay, they have a resting potential that is negative, right? So sodium is pumped out of the neuron, so the sodium-potassium pump is very important, right? And potassium primarily exists within the neuron, and so you have this difference, and this separation of charge, if you will, and your membrane potential tends to be more negative inside of the neuron compared to outside, just like myocardial cells, right? And if you had to imagine, do you think that takes a lot of energy to maintain that potential and to reset that potential constantly?
Yes.
Yes. So guess what? Neurons, just like cardiac tissues, require large amounts of ATP, which means they use lots of oxygen, which means, just like cardiac tissues, are they going to be particularly resilient to the effects of ischemia? No. In fact, they are even less resilient than myocardial cells are, okay? So nervous tissues tend to be highly susceptible to hypoxia, okay? You guys, and that kind of makes sense. They require lots of oxygen, lots of energy is produced. Yeah, okay, cool. So what happens here? You have all of these dendrites. You guys okay with that? All these dendrites here? And some of the information that comes into the dendrites will make the neuron less likely to depolarize, and some of the information will make the neuron more likely to depolarize, okay? And information that makes the neuron less likely it creates what's known as an inhibitory presynaptic potential. And this type of information tends to cause chloride channels to open. And when chloride channels open and chloride enters the cell, what happens to the membrane potential of the neuron? It decreases, right? Which makes it more or less likely to depolarize. Less likely. It's harder to depolarize. It is hyperpolarized. Does that make sense? You guys okay with that? All right. Now, excitatory presynaptic potentials tend to cause sodium channels to open. And when sodium enters the neuron, does the potential become more or less positive? More positive, and becomes easier for the neuron to depolarize, right? It moves toward the threshold. Just like cardiac cells, neurons also have a threshold potential. Are you guys okay with that? And what happens is, once the threshold is met, large amounts of sodium channels at the axon hillock open up, and sodium rushes into the cell and causes the depolarization to occur. Make sense? And so what happens is, you've got, 10, 20,000 different little connections at the neuron here, right? So you have massive amounts of information coming into the neuron, some telling it to depolarize, some telling it not to depolarize. And what happens is when the sum of all the excitatory and inhibitory information reaches the threshold, okay, so when at the end of the day, okay, so basically when the positive information, the information that favors depolarization wins out, what happens to the neuron? The whole thing depolarizes. Does that make sense? Okay, the sodium channels in the hillock open and the entire neuron depolarizes. Are you guys okay with this? And this is known as temporal summation. It is a summation of the inhibitory and excitatory presynaptic potentials over time, right? That's what temporal means, over time. So over time, when the excitatory potentials, or sometimes these are known as graded potentials, okay? Because they're small little bumps, right? But when enough of the small little bumps add up to get to the threshold, then you have an action potential occur. You guys okay with that? Okay, cool. So what happens with this action potential is it travels down the axon. You guys okay with that? And so you have all the sodium that rushes in at the axon hillock, and then what happens? Those sodium ions diffuse out, right? And they move from an area of high concentration to an area of low concentration down the axon. You guys cool with that? But what happens to these sodium ions as they move down the axon? Well, they start to spread out. You guys see how these little arrows? And so that signal becomes weaker and weaker and weaker, right? And so if you had the entire axon covered in myelin like this, what would happen is that signal would spread out and it becomes so weak and diluted that once it got to the other end, if it got to the other end of the axon, it wouldn't be able to trigger release of neurotransmitter. So what happens is every so often you have a break in the myelin and you have a new myelin sheath and you have what's called the node of Ranvier, and at the node you have sodium channels. And so as that signal weakens out, once it gets to a node, it causes these sodium channels to open, more sodium rushes in, and it quote-unquote recharges the signal. It amplifies the signal. And then the signal passes through the next section, it becomes weak, it gets to the next node, it gets amplified, and so on and so forth. And so you kind of have like a hop-skipping kind of thing going on where you have sodium channels, sodium channels, sodium channels, and that hopscotch or that hop-skipping kind of motion is what's known as saltatory conduction. Are you guys okay with that? And does that kind of make sense why we need that to happen? Because we need to amplify that signal. And again, an axon can be very long, guys, meters in length. Okay, there are some axons that basically go all the way from your central nervous system all the way down to your big toe. Okay, one single axon from a neuron. So as you might imagine, you need to have lots of nodes, okay, to ensure that that signal can get down to the end. And what happens once that signal gets down, and here we are down at the axon terminals? Well, that signal acts upon voltage-gated calcium channels. And calcium rushes into the cell, and calcium interacts with these vesicles full of neurotransmitters and allows these neurotransmitters to attach to the inner membrane and to merge with the membrane and open up and release and dump the neurotransmitter molecules out into the synapse. Okay? You guys okay with that? Okie dokie. So what I want to do is I just want to ask you guys a couple of questions. Do you think that we could make use of drugs that perhaps interact with inhibitory receptors?
Yes.
Yeah. And there are two classes of drugs that I want to mention that interact on something known as a GABA receptor. GABA stands for gamma aminobutyric acid. And GABA is actually an inhibitory neurotransmitter. And GABA receptors are what are known as ligand-gated ion channels. They belong to the ligand-gated ion channel family. And so a GABA receptor, when it is activated, opens up a chloride channel and allows chloride to enter the neuron and makes it harder for that neuron to depolarize. You guys okay with that? And two drug classes that actually interact with these GABA receptors are what we call benzodiazepines and barbiturates, like phenobarbital. Benzodiazepines are things like diazepam, midazolam, and lorazepam. And what do we use these benzodiazepines for clinically? Seizures, right? That's one of the primary uses in pre-hospital medicine, is to control seizures with benzodiazepines, right? And that's how they work, is they increase chloride transport into the cell that hyperpolarizes the neurons and makes it more difficult for the neurons to depolarize, which causes depression of the central nervous system. You guys okay with that? Now, too much, obviously, can depress breathing centers and cause problems there. But are you guys okay with how that works? Okay, and you guys should be familiar with your doses of major benzodiazepines, particularly when it comes to seizure control, right? If not, please just review, particularly lorazepam, diazepam, and midazolam. Those are your three major benzodiazepines. Now, because sodium channels, just like in cardiac tissues, sodium channels are an important part of depolarizing, do you think drugs that block sodium channels may affect the central nervous system and the peripheral nervous system? Profoundly, okay? So sodium channel blockers such as lidocaine, what do you suppose they're going to do?
Stop the ability for them to reach that threshold.
They're going to prevent, well, they not even the threshold.
Can't depolarize.
Yeah, but they will simply prevent that neuron from depolarizing, right? And where might these play a very important role, these sodium channel blockers?
Anesthesia.
Local anesthesia, right? If you want to suture a wound, what do? We'll inject lidocaine in around that area. And what does the lidocaine do? It prevents these sodium channels from opening up in the neurons, prevents them from depolarizing, and prevents the afferent neurons from sending pain information up to the central nervous system. Does that make sense? Now, sometimes you could actually overdose somebody on lidocaine if you gave them large doses IV, and that lidocaine could poison their central nervous system. And paradoxically, it could actually make them more prone to things like seizures and tremors, right? And isn't that a sign of potential lidocaine toxicity? Is somebody who develops altered mental status and may even go on to develop seizures. You guys okay with that? Okay. Yes, indeed. So far, so good? All right, we'll move right along. Let's see. You know what? I'm going to stop here since this is kind of a weighty discussion. I'll stop here, get you guys on a break.